Welcome, Leslie. We're so pleased to have you here from Indiana, one of our favorite places for type 1 diabetes research. I'm just going to introduce you, give a short bio. Leslie Wagner is a PhD candidate, and she's just about ready to defend. She's put together some phenomenal work, and they have a fantastic paper that's just come out in a preprint in bioRx. So would advise everyone to take a look at that. I'll dump that in the chat shortly. The title of her talk is Beta Cell Heterogeneity in the Acute Interferon-alpha Response. A little about her. She's a PhD candidate, as I mentioned, in Linnemann Lab at Indiana University School of Medicine. Her research focuses on using advanced microscopy techniques to study how pancreatic beta cells respond to cytokines produced during the innate immune response to viral infections, specifically evaluating reactive oxygen species or ROS production. And she will, as I mentioned, she's defending next week. She's eager to pursue a career in scientific writing, and she enjoys spending time with her two puppies outside of the laboratory. So thank you so much for joining us, Leslie. I'm going to drop this paper link into the chat, and everyone should feel free to post questions in the chat, and we'll also take questions at the end.
Thank you so much for this opportunity to present my work today at Sugar Science. My name is Leslie Wagner, and I'm a PhD candidate at the Indiana University School of Medicine in the lab of Dr. Amelia Linnemann. And as Monica said, I am set to defend my PhD next Friday, so I'm really excited. And today I will be sharing some of my thesis project, which is currently under review and is available on bioarchives. So as everyone here probably knows, type 1 diabetes is a multifactorial disease with both a genetic predisposition and an unknown environmental triggering event that occurs before clinical diagnosis. And although still somewhat controversial, the leading hypothesis for this environmental trigger is viral infection. So during viral infection, the immune system responds by producing various cytokines and interferons, such as interferon alpha. Other pro-inflammatory cytokines have been shown to induce reactive oxygen species or ROS production, as well as ER stress and DNA damage. It's important to note that ROS are beneficial signaling molecules to the cell. However, when levels increase above the capacity of scavenging enzymes to mitigate the stressor, oxidative stress and cell death can pursue. And so how does our lab study ROS in the beta cell? So we take human islets and transduce them with an adenovirally packaged GRX1 RoGFP2 biosensor. So this biosensor has been well established in other organs, such as the liver. But what we did is we put this biosensor under the RIP1 promoter to make it beta cell specific. And it's important to note that this biosensor is ratiometric and reversible, making it ideal for in vivo studies. And we take these transduced islets, transplant them under the kidney capsule of immunodeficient NSG mice, and then 2 weeks following transplantation to allow for optimal sensor expression and vascularization, we externalize the kidney containing grafts on an imaging dish and conduct intravital imaging while the mouse is anesthetized. And during intravital imaging, we can incorporate various treatments such as interferon alpha on the stage. And so shown in figure 1A of my paper are representative images of those transplanted human islets expressing this GRX1 RoGFP2 biosensor. And shown in the top panel is an image collected at baseline, and then shown in the bottom panel is an image collected 60 minutes after interferon alpha administration. Highlighted with a white arrow is a cell that is producing ROS in response to interferon alpha, and we term these cells responders. And how we identified the cell as a responder is because after interferon alpha, there is an increase in the 405 signal shown in magenta and a subsequent decrease in 488 signal shown in green. And cells that lack this response, we term non-responders. So then in figure 2B, we represent how this biosensor works. So in the presence of ROS, this biosensor becomes oxidized and becomes more excitable at 405 nanometers. In figure 1C, we show representative heat maps from an islet in a mouse that was stimulated with saline or interferon alpha. And here we show individual beta cells throughout the time of imaging, and represented in the middle is the full change in the 405/488 ratio of the biosensor compared to the baseline for each time point. And what I would like everyone to appreciate is that in the interferon alpha stimulated islets, a subset of beta cells produce reactive oxygen species beginning around 12 minutes, and this response is sustained throughout the time of imaging. So shown in figure 1D is the full change in ratio of the biosensor for all individual beta cells from all donors. And I have chosen some representative time points for imaging, and this shows that about 10 to 20% of all beta cells during these time points have this response in the interferon alpha treated group, with the peak response being around 45 minutes. And so we further investigated the percent responders from each individual donor, and we found that some donors have no responders, shown here, and some have as high as 60% responders. And so to investigate this inter-donor heterogeneity, we turned to the human islet phenotyping program provided by the Integrated Islet Distribution Program and plotted various variables for each donor. And when we analyzed the correlation between each donor's percentage of responders, we observed a strong correlation with two variables. And interestingly, these were both strong negative correlations with BMI and HbA1c. And this suggested to us that healthier donors or donors with lower BMI and lower HbA1c values actually have more cells that produce ROS in response to interferon alpha stimulation. And so in figure 3, after identifying that interferon alpha stimulates ROS production in a subset of human beta cells, our next goal was to identify the specific type of ROS being produced in these cells. And so as shown in figure 3A, we treated and stained healthy human islets in vitro with hooks to visualize nuclei as well as dihydroethidium or DHE to measure superoxide production in these islets. And in the presence of superoxide, DHE undergoes a conformational change, allowing it to intercalate with nucleic acids such as DNA, resulting in a strong nuclear stain when superoxide is present. And so we use antimycin A here, an uncoupler of the electron transport chain, as a positive control. And then shown here in this lower panel, we alternatively stain human islets with DCFDA, which is a fluorescent probe used to measure hydrogen peroxide in the cells. And so we analyzed this data shown here to the right. With DHE, we looked at both the percent positive nuclei and the number of positive nuclei per islet micron squared. And we found a significant heterogeneous increase in both of these measurements following interferon alpha stimulation. However, when we quantified the DCFDA intensity as mean intensity of signal, we did not find a significant difference. However, there was a trending increase in hydrogen peroxide production that could be explained by the fact that the first step of superoxide metabolism is conversion to hydrogen peroxide by the superoxide dismutase enzymes. And so lastly, I repeated this experiment with the dihydroethidium in islets from donors with type 2 diabetes and did the same quantification as number of nuclei per micron squared, as well as the percent positive nuclei. And I see that this response is diminished, which correlates well with the observation that we had in vivo, that donors with higher BMI and higher HbA1c values have less response. And so next, we wanted to identify transcriptional network differences that predisposes a specific population of islet cells to ROS in response to interferon alpha. So we acutely treated human islets from 3 human donors with vehicle and interferon alpha for 1 hour. We were first interested in comparing the differences between the vehicle treated cells exhibiting basal ROS production and the responder population to identify unique effects of interferon alpha stimulation on ROS producing cells. So this is shown in figure 4A, where we dissociated treated islets and stained them with DHE and flow sorted into populations both negative and positive for ROS. I don't show it here, but we used antimycin A as a positive control for gating. And so then we collected RNA from these samples and conducted bulk RNA sequencing. And so shown in figure 4B, we did an analysis and identified 296 upregulated and 386 downregulated genes. And then we next performed gene ontology analysis with these upregulated genes and found a lot of interesting results, such as increased expression of several antiviral and immune-related pathways, including MDA5 and RIG1 signaling. And notably, both MDA5 and RIG1 themselves were significantly increased in the responder population. And so once we determined the unique effect of interferon alpha on ROS-producing human islet cells, we next wanted to identify differences in gene expression of human islet cells that produce ROS in response to interferon alpha stimulation from those that don't, even when exposed to interferon alpha. So therefore, we compared differentially expressed genes from the non-responder and responder populations that I've shown here in figure 5A. And given the acute time point, this should tell us about the transcriptome that predisposes these cells to ROS production. And so shown in figure 5B, analysis revealed over 1,000 upregulated and downregulated genes between these populations. And when we conducted gene ontology analysis, it revealed several upregulated pathways, including immune response and inflammatory response in the responder population, as well as a decrease in apoptosis pathways, which was interesting. And so since this analysis was conducted on whole islet cells, we first wanted to identify if these cells were enriched in any specific islet cell type. And so we show in figure 5D that the responders are enriched in several beta cell identity markers and have lower expression of glucagon, suggesting that this population is enriched for beta cells. And since ROS levels are mitigated in the cell by the antioxidant response, we next investigated the expression of various antioxidants between the non-responder and responder populations. And we found decreased expression of several antioxidants, including superoxide dismutase 2, which is a mitochondrial specific antioxidant for superoxide. And then we also investigated several key survival and stress response genes between these populations, and found that interferon alpha responders showed increased expression of genes linked to cellular stress responses, including ER stress, inflammation, and mitochondrial quality control. Interestingly, they also had decreased expression of all the HLA class 1 genes, which are associated with T-cell mediated beta cell destruction and type 1 diabetes. And lastly, I wanted to note that we did not see differential expression of either interferon alpha receptor between the responder and non-responder populations, suggesting that this differential response is not due to differences in receptor availability. And so to investigate if there was any relationship between these differentially expressed genes in the responder population and disease state, we compared the genes with single cell sequencing data provided to us by the Human Pancreas Analysis Program. And this was in collaboration with the Kessner Lab at the University of Pennsylvania. And they conducted gene set enrichment analysis between the upregulated genes and the responder population and beta cell, single cell sequencing data from donors with type 1 diabetes and respective healthy controls. And shown here in figure 6A, this analysis revealed that genes upregulated in the responder population were significantly enriched in genes with high beta cell expression in the healthy controls. And then we show a heat map of these leading edge genes in figure 6B in case people were interested in these genes. And lastly, gene ontology analysis of these leading edge genes that we show in figure 6C revealed pathways involved in ER stress and protein folding. And overall, this suggested to us that interferon alpha responders may be greatly reduced or selectively lost during type 1 diabetes. And so lastly, to determine if the cytoplasmic ROS increase in response to interferon alpha originates in the mitochondria, which is an organelle known for ROS production, we first stained endo-C cells with mitosox green and confirmed that interferon alpha stimulates mitochondrial ROS production that we show in supplementary figure 4. But then we were interested in further defining the timing of this ROS generation. And so we transfected endo-C cells with Rho-GFP2 based biosensors targeted to either the cytosol, which is the RIP1 GRX1 Rho-GFP2, or the mitochondria, which is the Th1nk GRX1 Rho-GFP2. And we treated these cells with interferon alpha and corresponding controls for 1 hour. And we see that interferon alpha induced a significant increase in ROS at 15, 30, and 60 minutes in both the cytoplasm, which I show here on the left, as well as the mitochondria shown here on the right. And for this analysis, we analyzed the fold change of the 405/488 intensity and quantified that from the average vehicle ratio. And we plotted individual beta cells for this experiment. And so these findings suggest that interferon alpha triggers mitochondrial ROS production, which is rapidly released into the cytosol where we observed it in vivo with intravital imaging. And so in summary, we have demonstrated that interferon alpha causes heterogeneous ROS production in healthy human beta cells, both in vivo and in vitro, in a subpopulation of cells that we term responders. And this observed ROS is superoxide that originates first in the mitochondria. We found that healthier donors or donors with lower BMI and lower HbA1c values actually have a higher proportion of these responders. And interferon alpha itself stimulates transcriptional expression of antiviral pathways that promote mitochondrial ROS production. And responders have a unique molecular signature consisting of increased expression of genes involved in immune and inflammatory response. And then we showed that the genes elevated in this responder population are enriched in the islets of non-diabetic donors, suggesting that responders are selectively lost during disease progression. And so I want to thank my lab for their continued support throughout my PhD journey, especially my mentor, Dr. Amelia Linnemann. I additionally want to extend my thanks to the Kestner and Brosink labs for their contributions to this work. I would like to acknowledge our funding and islet procurement through both Alberta and the IIDP. And lastly, perhaps most importantly, I want to extend my sincerest gratitude to the organ donors and their families for their precious donation that makes this work possible. And I will be happy to take any questions. Thank you.
Thank you so much, Leslie. That was phenomenal. Really great work for a PhD thesis, truly. I wanted to open up for questions, some clapping in the audience, so that's fantastic. Any questions? Feel free to unmute yourself.
Hey, I have one.
Yes.
Okay. Hi, Leslie. It was a nice talk. I came a little late to the talk, so maybe I missed this part. Just wanted to clarify if this particular cytokine or any cytokine cocktails, do you see any changes in ATP levels? Have you measured ATP at all?
No, we have not measured ATP at all.
I see. Okay. Just wanted to see if it has got to do with anything to do with that ROS generation. Does it affect the OX4s or anything?
Yes,
So,
yeah, thank you so much. That's a great question and something that we definitely should investigate.
okay, thanks.
Any other questions from the audience? Not yet. I have a question. So if you have, if you have this situation with a subpopulation of the beta cells, do you have any evidence, or have you even anecdotal evidence that you've seen where this actually happens in other cell populations that make up the islet, like the alpha cells, for instance, or other cells?
So no, I have not seen any literature with that. About the mitochondrial ROS production in response to interferon alpha, though, there have been studies in both airway epithelial cells as well as glial cells that show that interferon alpha stimulates mitochondrial ROS production in those cells to then promote a more beneficial antiviral response. And so to my knowledge, this is the first time showing that this response also happens in the islet, specifically in the beta cell.
Yeah, I did a little sort of back research and saw that, neutrophils, endothelial cells, and some cancer cells also show this heterogeneous response. So, but it's really fascinating that you've got it in the beta cells. So that's like, one subset of the islet cells. And so that's really fascinating. I wondered also, if you wanted to comment a little bit. I know another student in the laboratory, Matt Austin, he's working on defective autophagy. How might, what you've uncovered here and his work maybe dovetail, and you don't have to give away everything, but maybe just like a short comment.
Yeah. So autophagy is activated in response to ROS. And so our lab has been shown that autophagy is maladaptive in type 1 diabetes And so this suggesting that responders are selectively lost in type 1 diabetes with our HPAP gene set enrichment analysis. It could be that this defective ROS production and the defective autophagy both promote this selective loss in this population of beta cells.
Yeah, exciting times coming from the Linnemann Lab. We can't wait to hear that story progress too. When you talk about a small subpopulation of uncharacterized beta cells that are innately resistant to immune attack, right? Do you see that across samples, that percentage or whatever the small size stays similar or the same, or is it very variable?
No, it's variable. So I show that, I think, in figure 2, where we look at the individual donor response and then plot the percent responders with the variables provided by IADP. And it's a very strong negative correlation. So the lower the BMI, the lower the HbA1c, the higher proportion of responders in that donor.
If we flip it around and think about type 2 diabetes, you've got the low BMI, right, and the low A1C. What if we flip it and think about it in context of type 2? Has anyone looked at that, or what might you expect?
Yeah, so I show a little bit of that in figure 3, where I treat and stain human islets in vitro with the dihydroethidium or DHE dye to measure superoxide production. And the healthy control islets with interferon alpha stimulation, there is a heterogeneous increase in ROS positive cells. However, with the type 2 diabetic islets, that response is ablated. And so it's really interesting and correlates well with what we see in vivo, where the donors with the higher BMI, the higher HbA1c actually had less response, and most of them had no response at all.
Interesting. Here's a question from City of Hope, Jemming Lu. Thanks for your great talk and work. I'm wondering how ROS production is affected by long-term interferon alpha treatment, such as over 24 hours. And additionally, does ROS show a similar response pattern to interferon? gamma?
Yeah, so I will answer the first question and say that Dr. Isrik has done a lot of long-term interferon alpha treatment studies. My thesis work is really only acute interferon alpha treatment, so I can't personally speak to that. And then with the second question, we have done experiments with interferon gamma. I believe experiment figure 6 of the paper highlights this, where with the looking at cytoplasmic and mitochondrial ROS production, I did include interferon gamma as a treatment and did not see the same response. So I can't say that it's interferon alpha specific, but I can say that interferon gamma does not appear to stimulate this.
Thanks for that. Sort of like clinical implications. Do you think there might, could this subset of beta cells with the heightened ROS responses serve as early indicators or maybe even a therapeutic target in a screening panel or something along those lines? Have you guys discussed this in the lab, or what are your thoughts?
Yeah, so I believe it does have implications identifying what makes this cell population unique and maybe have a therapeutic strategy to target those cells and potentially stop this ROS production, because it appears that these cells are selectively lost in disease. And so that is definitely something worth exploring.
Yeah, it's kind of interesting. Anyone else? I did want to talk about the actual, you have a great core there at Indiana, and some of the optical work that you did there, the imaging work it's featured, or it's housed in that particular core, correct?
Yeah. So Amelia will answer that.
Yeah. So
Oh, thank
yeah,
you.
sorry, I'm sitting here. So yeah, I run our microscopy core for the Diabetes Center, where we're doing a lot of imaging, not just intravital imaging. We're also doing a lot of codex imaging, and imaging other tissues. So we do pancreas, we do transplants of islets in the kidney capsule, predominantly human islets, although we have begun to do some work with iPSC-derived islets-like cells as well. And we are working on building additional collaborations in that realm. So we do have some other outside investigators that have sent us animals that we've done imaging on, and we've published some of that work. But for those that are listening that are interested in this, we are also open to new collaborations within our core. Ultimately, we want to be able to share the work that we're doing and the techniques we've developed with the community to solve problems.
Yeah, fantastic. I did want to shine a little light on that, because I've just heard so many great things about the technical expertise that is sort of living there at that core in Indiana. So is there any last questions? Let me look through this. I don't see anybody else. I think this is just a phenomenal new approach, and thank you so much for sharing with us. Good luck next week in your defense, and I hope you have a great career in scientific writing. But I also would like to give a shout out to this lab. This Linnemann Lab has really some fabulous work going on, and if there are people who are graduate students, new graduate students, or postdocs interested, this is really an excellent place to do some fabulous work. So think about it and explore it and reach out.
Thank you, Monica.
Thanks again, Leslie.
Thank you very much.
Good to talk to you. Best of luck. Bye-bye.